Hello, and a very warm welcome to the Art Biolab channel. Have you ever considered the possibility of putting life on pause? Just like in the movies, stopping time for living cells and then bringing them back perfectly preserved weeks, months, or even years down the line? Well, you might be surprised to learn that this isn't science fiction at all. It's a real, tangible process happening every single day in laboratories all across the globe. Today, we're going to delve into the incredible art of suspended animation.
So, that's the big question, isn't it? How is this even possible? How can you take something as fragile, as delicate as a living cell, freeze it solid without utterly destroying it, and then successfully bring it back to life? The answer, it turns out, lies in a fascinating and incredibly precise technique known as cryopreservation. And that's what we're here to do, break down exactly how scientists pull it off.
Now, to really grasp why scientists needed to master this art form, we first have to understand the fundamental challenge, the core problem that every single cell culture lab faces. Just try to imagine it. You're responsible for keeping millions upon millions of tiny living organisms alive and well day in and day out. It's a relentless, high-stakes operation with a constantly ticking clock.
You see, continuously growing cells, a process that scientists call passaging, it's not just a difficult task, it's an unbelievably risky gamble. You're in a constant fight against time, against biology itself, and of course, against the budget. It's a totally unsustainable model, one that puts vital, sometimes irreplaceable research in constant jeopardy. So, what exactly are these dangers we're talking about?
Well, this really lays out just how many things can go wrong. First off, microbial contamination. A single stray bacterium or fungus can get in and completely ruin an entire experiment that took weeks to set up. Then there are the cells themselves. Over time, they can just change. They can undergo genetic mutations, or they can lose the very characteristics that you were trying to study in the first place. You also run the risk of cross-contamination, where one cell line gets mixed up with another. The cells can even just get old and stop dividing. That's a process called senescence. And if all that wasn't enough, it is a massive drain on expensive resources and precious time.
So, confronted with this relentless ticking clock, scientists came up with a truly elegant solution. Instead of trying to force these cells to run a biological marathon they were almost guaranteed to lose, they figured out something much smarter. They figured out how to hit the pause button, stopping the race altogether until they were ready to pick it up right where they left off.
And this brilliant solution is what's known as a cell banking system. The best way to think about it is like a biological library, or maybe a living time capsule. What scientists do is create a master stock of their cells, freeze down dozens and dozens of vials, and then store them safely in liquid nitrogen. So whenever they need a fresh, pristine, completely unchanged sample for a new experiment, they can simply go to the library and check one out.
Okay, but that brings us back to the how. How do you actually freeze a cell without killing it? I mean, we all learned in school that when water freezes, it expands. The ice crystals that form inside a cell can act like millions of microscopic shards of glass, just tearing the delicate cell membrane to pieces. The success of this entire incredible process hinges on one simple but absolutely critical rule.
This right here, this is the mantra, the golden rule of every cell culture laboratory in the world. Slow freezing, rapid thawing. It might seem a little counterintuitive at first glance, but I promise you, this simple phrase is the secret key to bringing cells back from the brink of absolute zero. So, why? Why does this specific combination work so well?
Okay, here's the crucial part. By freezing slowly, you give the water inside the cell enough time to seep out through its membrane before it can turn into those lethal internal ice crystals. This is the whole game. The water ends up freezing on the outside of the cell, which is far, far less damaging. Then, when you're ready to bring them back, you do the exact opposite. You thaw them rapidly. That in-between phase where ice is melting and recrystallizing is actually an incredibly dangerous time for the cell. A quick plunge into a 37-degree water bath gets it back to a safe, stable temperature in just a minute or two, dramatically minimizing that potential damage.
And believe me, when we say slow, we mean incredibly, painstakingly precise. The optimal cooling rate, as you can see, is just 1 to 3 degrees Celsius per minute. This isn't just a matter of tossing a vial into a standard freezer. Oh no, it requires very specialized equipment that can control this gradual, gentle descent into a frozen state, making sure the cells survive the journey intact.
But, as it turns out, a controlled temperature is only half the battle. To truly protect these cells on their journey into the deep cold, scientists need to give them some help. They add a special protective cocktail. Think of it as a kind of cellular antifreeze.
And preparing the cells to receive this cocktail is a vital first step in the whole process.
Right. You can't just freeze any old cells. You have to choose the absolute best of the best. This is basically a pre-flight checklist for the cells. They have to be healthy, they have to be young, and they need to be in what's called the logarithmic growth phase. That means they're actively and happily dividing. You also need to confirm that their viability is over 90%. You even give them fresh food, a new culture medium, about 24 hours beforehand, just to make sure they're in peak physical condition. Only the strongest candidates are chosen for this journey.
Now, the key ingredient in that antifreeze cocktail we mentioned is a special class of chemicals called cryoprotectants. Their job is really twofold. First, they help to draw that water out of the cell, and second, they physically interfere with the formation of ice crystals. You can think of them as a molecular shield, protecting the cell from freezing damage.
And in the world of cryoprotectants, there is an undisputed champion. It's a substance called dimethyl sulfoxide, or DMSO for short. What makes it so special? Well, because it's a very small, fat-soluble molecule, it can quickly and easily slip right through the cell's membrane. This allows it to protect the cell from the inside out. And it's this unique property that makes it the go-to standard choice for the vast majority of cell lines.
Of course, in science, it's rarely a one-size-fits-all situation. As you can see from this table, DMSO isn't perfect for every single case. For certain, very specific cell lines, like one called HL60, DMSO can actually trigger the cells to change into a different cell type, a process called differentiation. And that's something you definitely don't want to happen. In those special cases, scientists will turn to alternatives like glycerol.
So the final recipe is this carefully balanced mixture. It's mostly the standard growth medium. You've got about 10% DMSO, and then a surprisingly high concentration of serum, often more than 20%. But why so much serum? That seems like a lot, right? Well, the DMSO, while protective, can make the cell membrane just a little bit leaky. All of that extra protein from the serum helps to stabilize the membrane, reinforcing it and making sure nothing important leaks out during the extreme stress of the freezing process.
All right, let's zoom out for a moment. We've talked about the problem, we've covered the golden rule, and we've broken down the antifreeze cocktail. This technique, which has been perfected over decades of work, isn't just some clever lab trick. It is, without exaggeration, a fundamental pillar that makes all of modern biological research possible.
Think about it. By mastering this art of pausing life, scientists effectively solved all those problems we discussed at the beginning. They can prevent contamination. They can completely stop genetic changes in their tracks. They can guarantee, with certainty, that the cells they use in an experiment today are genetically identical to the ones they'll use 5 or even 10 years from now. This provides the consistency, the reliability that is the absolute bedrock of all good science.
So right off the bat, why can't you just toss a vial of cells into your freezer at home? The answer, in one word, is ice. See, when water freezes slowly, like in your freezer, it forms these big, jagged ice crystals. Now, imagine those inside a delicate little cell. They basically act like tiny knives, just shredding everything. Membranes, organelles, it's lethal damage.
And you've got to understand, the stakes here are incredibly high. We're talking about cell lines that are maybe irreplaceable, used for cancer research or developing new drugs. Think about banking stem cells for future therapies. The entire point of this whole complicated dance is to get these cells into a deep freeze and back out again, alive and ready to go.
Okay, so before you even think about freezing anything, you have to become a bit of a coach. You're getting an athlete ready for the biggest game of its life. The journey into the cold is unbelievably stressful for a cell, so only the absolute healthiest, most robust cells even get a chance to try. And this is the golden rule. Cell viability, that's just the percentage of living, healthy cells in your sample, it has to be above 90%. Why so high? Well, because you're going to lose some cells during the whole freeze and thaw cycle. It's just inevitable. So if you don't start with a super strong and healthy population, you won't have enough good cells left on the other side.
And that leads us to this pre-freeze checklist. It is absolutely non-negotiable. First, you check for contamination. You don't want to perfectly preserve a bunch of bacteria, right? Then, you make sure the cells are in their logarithmic growth phase. That's basically their happy place, their peak moment of health and division. And giving them fresh food, or medium, about 24 hours before gives them one last boost of nutrients for the long trip ahead.
All right, our cells are prepped, they're healthy, they're ready to go. Now we get to the main event, the actual, precise protocol for guiding them safely into that deep sleep. Let's walk through this real quick. First, you gently detach the cells from the surface of the flask where they're growing. Then you use a centrifuge to spin them all down into a tight little pellet at the bottom of a tube. You get rid of the old liquid, and here's the key part. You resuspend that pellet in a special protective freezing medium. Finally, you get the concentration just right, making sure each little vial has millions of cells, all ready for the big freeze.
Now, this might sound like a no-brainer, but I really can't overstate how critical good labeling is. Just imagine thawing a vial 10 years from now and having zero idea what it is, when you froze it, or how many cells are supposed to be in there. It's a complete waste. Meticulous, obsessive record-keeping is just the bedrock of good science.
So I mentioned that special freezing medium a second ago. What's in this magic stuff? What's in this survival kit? Well, it's a really carefully balanced cocktail, and it's designed to protect those cells from all the dangers of the cold.
The star of the show is an ingredient called a cryoprotectant. Honestly, the easiest way to wrap your head around it is to just think of it as biological antifreeze. Its whole job is to lower the freezing point of water and, most importantly, stop those big, deadly ice crystals from ever forming inside the cells.
So this is the standard recipe, and it's pretty interesting when you break it down. 70% is just basic cell food, the stuff they're used to. 20% is fetal bovine serum, or FBS, which is just packed with proteins and growth factors that help cells handle stress. And that last 10%, that's our star cryoprotectant, DMSO. Each part has a really important job to do.
But here's a classic example of how science is never one size fits all. That DMSO we just talked about, well, for some really sensitive cell lines, like one called HL60, it can actually be toxic. It can make them change into something else entirely. In cases like that, scientists have to get creative and swap out the DMSO for a gentler option, something like glycerol. Okay, our cells are prepped. They're protected in their little cryo cocktail and they're sealed up in their vials. It's time for the final two-stage journey into the ultra-cold, where they're going to wait for whatever mission the future has in store for them.
So how do you pull off that perfect, slow cool down? Well, there are two main ways to do it. The simplest is a special box filled with isopropanol. You just stick it in a -80 freezer and it does the work for you, guaranteeing a slow, steady drop in temperature. For the big labs that do a ton of this, there are programmable freezers that give you incredible precision, but yeah, they come with a pretty hefty price tag.
And this is the magic number. The perfect cooling rate for most cells is -1 degree Celsius per minute. Go any faster, you get those ice crystals inside the cell. Go any slower, and the cell can literally die of dehydration as water gets pulled out of it. It's this super delicate balance, and hitting that rate is the real secret to success.
So here's that two-stage process in action. Step 1 is that controlled overnight freeze down to -80 Celsius. But you know what? That's just a temporary stop. For real long-term storage, where basically all biological activity just stops, you need a quick transfer into the insane cold of a liquid nitrogen tank. We're talking about -196 degrees Celsius. Unbelievably cold.
And, success isn't just about the fancy science. It's also about good old-fashioned discipline. You have to log exactly where every single vial is stored. You have to have dedicated spaces so someone doesn't accidentally move your samples halfway through the process. I love this last point. It even notes that, in a pinch, a thick-walled styrofoam box can work as a substitute for a proper freezing container. That's some real scientific ingenuity right there.
What if I told you that we can literally press pause on biological time? That we can stop life in its tracks and keep it that way pretty much indefinitely? It sounds like something out of a sci-fi movie, right? Well, it's not. And it all comes down to one single, incredibly cold number.
Yep. -196 degrees Celsius. That's the temperature of liquid nitrogen. And when you get that cold, the normal rules of biology, well, they just don't apply anymore. So here's the wild part. Once you get below about -135 degrees Celsius, all the metabolic activity inside a cell just stops. It grinds to a complete halt. It really is like hitting the pause button on life, allowing us to preserve cells for decades. But to get to that permanent pause, labs have to make a pretty fundamental choice.
So what's it going to be? Do you go with a super complex, sophisticated machine, or do you choose a substance that feels like it belongs on a spaceship? This one decision, it impacts literally everything, from the cost of running the lab to the safety of the samples themselves.
And this is really the heart of the trade-off. On one hand, you have electric freezers. They're way easier on the budget day-to-day, but they're also mechanically complicated, and you better have a good backup power source. On the other hand, you have liquid nitrogen. Mechanically, it's super simple and gets you to that extreme -196 degrees, but it's expensive and needs to be refilled constantly. Oh, and it comes with one major, kind of hidden risk.
And this quote just nails the danger.
See, if a vial isn't sealed absolutely perfectly, the liquid nitrogen itself can become a highway for contamination. It allows viruses and pathogens to travel from one sample to another. It's a tiny risk for sure, but with absolutely catastrophic potential, and that very risk is what led scientists to find a compromise.
So faced with that problem, scientists got pretty clever. They figured, how can we get the incredible cold from liquid nitrogen without the liquid ever touching the samples? The solution is to use the gas, the vapor that comes off the liquid. It's called the vapor phase.
Here's how it works. The vials sit on these racks that are suspended above a pool of liquid nitrogen at the bottom of the tank. This creates what's called a temperature gradient. It's coldest down at the bottom, around -190 Celsius, and it gets a little bit warmer, up to -135 Celsius, near the top. So the sample stays safely frozen, but they never actually take a bath in the liquid. This completely eliminates that cross-contamination risk. But as you can imagine, maintaining this delicate balance requires some serious care, because any big temperature fluctuation could ruin everything. That means you have to keep the tank at least half full at all times, you have to limit how often that lid gets opened, and you absolutely have to have an alarm system to warn you if the nitrogen levels are getting low. In this world, stability is everything. And that stability doesn't just come from the machine, it comes from the system the people use to run it.
You should really think of it like a library for life itself. Every single one of those little vials is a book containing priceless information, and you need a flawless cataloging system to find what you need without disturbing anything else.
The number one goal of any inventory system like this is speed. You have to know exactly where a vial is before you even think about opening that lid. Why? Because every single second the tank is open, warm air rushes in, the temperature fluctuates, and the entire precious collection is put at risk.
A really solid system has layers of backups. You'll have a physical logbook, old school pen and paper, and a computer database. You need a strict protocol for checking samples in and out. And, of course, every single vial needs its own unique label. And it has to be a label that can survive that extreme cold without peeling off or becoming impossible to read.
And when it comes down to it, it's all about simplicity. You just need 3 key pieces of information on that label. What is this? What's its unique number? And when was it frozen? This simple label is the cornerstone of the entire system, making sure a sample's identity is never lost. But the biggest risk in the lab isn't losing a sample. It's the danger to the people handling them.
So we've talked a lot about the cells, but what about the humans? The people handling this incredibly dangerous substance every day? Because liquid nitrogen poses a silent, invisible, and potentially lethal threat.
The primary danger is something called asphyxiation. As liquid nitrogen evaporates, it expands into a huge volume of nitrogen gas, and it literally pushes all the breathable air out of a room. And the scariest thing is, it has no color, no smell, it gives you absolutely no warning before you could lose consciousness. This is the number that matters, 18%. Normal air, what you're breathing right now, is about 20 to 21% oxygen. Safety alarms are set to go off when the level in the room drops below 18%. That is a tiny, tiny margin for error, which is why every lab that uses this stuff needs incredibly strict safety protocols.
If that alarm goes off, the instructions are simple and they are non-negotiable. You get out immediately. You do not go back in for any reason. You wait until the ventilation systems have done their job and the oxygen level is back to normal. Seriously, there is no room for heroics here.
And this all leads to one simple critical rule. Never work with liquid nitrogen alone. The buddy system isn't just a good idea. It's a critical safety measure when the greatest danger you face is completely invisible.
At the end of the day, this technology is amazing. It allows us to pause biology, to store the very building blocks of life. But it demands this meticulous dance with extreme temperatures, invisible dangers, and of course, human error. And it forces us to ask a really big question. In this quest to preserve life, what is the right balance between scientific progress and absolute safety?